உங்களுக்கு ஆப் இருக்கா சார்? ஜூம் ஆப் இருக்கா?
ஆக்சுவலா கூகுள்
அங்க 2 பேர் வந்திருக்காங்க. ஸோ நீங்கள் ஜாயின் பண்ணும்போது ஜாயின் பை ப்ரௌசரே கொடுத்துட்டீங்கன்னா ஓகே சார். நெக்ஸ்ட் ப்ரொசீஜர் பார்க்கணும்னா இது எப்படி இருக்கும் சார்? நீங்கள் லான்ச் மீட்டிங் கொடுத்துட்டீங்கன்னா ஓப்பன் யூஆர்எல் ஜூம் ப்ரௌசர் லான்ச்சர் கேட்கும். ஜாயின் ஃப்ரம் யுவர் ப்ரௌசர் கீழே அங்கே உங்கள் என்ன பேரில் லாகின் பண்ணணும்னு கேட்கும். ஜாயின் கொடுத்துருங்க. மீட்டிங் பாஸ்போர்ட் கேட்கும். பாஸ்போர்ட் வந்து 217. உங்களுக்கு அந்த மெசேஜ்ல இருக்கும். நான் இன்னும் அனுப்பல இன்னைக்கு. HTML மட்டும் கொடுத்தீங்கன்னா போதும். உங்களுக்கு HTML
ஆக்சுவலி இது ஒர்க் ஆகுது. அந்த பின் டாட்
அந்த VJ உங்ககிட்ட VJ இருக்குல்ல. ஸோ நீங்க உள்ள வந்து அப்புறம் இங்க இது கூட வைஃபை பாஸ்வேர்டு நீங்க ஹெட்ஃபோன்ஸ் போட்டு எடுக்கிறீங்களா?
வாய்ஸ் ரெக்கார்டிங் போட்டு அப்படியே போயிடுமா சார்?
டைரக்டா போகும். பட் எப்படி இருக்கும்னு நீங்க செக் பண்ணிக்கோங்க.
இப்போ அப்படியே எடுக்கிறேன். ஒரு வேளை போகலன்னு சொன்னா நெக்ஸ்ட் கிளாஸ்
பேசுங்க சார்.
ஹலோ ஹலோ ஸ்டூடெண்ட்ஸ், கேன் யூ ஹியர் மீ? ஹலோ ஆக்சுவலி கேக்குது. ஹலோ ஸ்டூடெண்ட்ஸ், கேன் யூ ஹியர் மீ? Am I audible?
Yes. Wi-Fi மட்டும் கனெக்ட் பண்ணா உங்களுக்கு நெட் ஃபாஸ்டா இருக்கும். நீங்க PPT ஓபன் வச்சுக்கோங்க. ஃபர்ஸ்ட் டைம் வந்து இந்த டைம் ஆச்சு சார். ஜூம்ல போயிட்டு உங்களுக்கு ஆக்சஸ் பண்ணுங்க. Screenshot பண்ணிட்டு இது மட்டும் வச்சுட்டு இந்த ஓகே.
Thank you, sir. Okay.
அந்த பசங்க இங்க வருவாங்க. ஒன்ல இருந்து 9 இங்கே வந்து சாண்ட் ஃபால் அப்படின்னு வரும் சார்.
கொஞ்சம் கனெக்ட் பண்ணிட்டாங்க.
நம்பர் போடுங்க. டைம் வரல சார். டைம் ஒர்க் ஆகலையா? இல்லை இதில் போட சார் நம்பர்.
எங்க போய் கனெக்ட் பண்ணீங்க? உங்களோட ஹாட்ஸ்பாட் கனெக்ட் பண்ணிட்டா? சரிங்களா?
கனெக்ட்
Yes. Okay. So, good morning. So, can you all able to see the screen? Any issues? In case of any issues, please let me know. Yeah, settle fast. Okay. Shall we start? Yes. So, so we will be discussing about the organelles of the cell and more about its structure, how the structure of every organelle is there, and we will be focusing on the chief functions of the particular organelle in a cell, right? Already we have discussed about some of the organelles. Like first, we started with cell wall. Okay, cell wall in plants. Look at the structure. See, in the first few slides, we will be recollecting the things which we have already discussed. Okay, but here you can see the diagrammatic representation of the things which we have discussed earlier. And as we know, like a cell wall is a protective layer which covers the cell. And we know that there are primary cell wall and secondary cell walls. Okay, three important components are present in the cell wall. The two types are primary cell wall and secondary cell wall. And these are the tough portions, but still they have some kind of junctions within them called plasmodesmata and the pits. So look at the structure, like and the white portion is the cytoplasm of the cell, and this layer is actually the cell membrane, and you can see the pink portion. That pink portion is the secondary cell wall, and the brown portion is actually the primary cell wall. Okay, cytoplasm, cell membrane, secondary cell wall, and primary cell wall. Okay, and in between the two cells, there is a gap junction, which is called what? Plasmodesma or plasmodesmata. Okay, and also there are, if you can look into the image, you can see like the brown portion is the secondary cell wall, which is made up of what? Which is lignified with means lignin and suberin, two important molecules which make the secondary cell wall. And you can see there are a small opening porous structures are there, which are called what? Pits. Okay, so this is about the cell wall in plants. Okay, primary cell wall, secondary cell wall. Primary cell wall primarily made up of cellulose, hemicellulose, and pectin. Secondary cell wall made up of lignin and suberin. The junctions which are present in primary cell wall are called plasmodesmata, and the secondary cell wall, a small porous structure, is called pits. Okay, so this is about cell wall in plants. And second, cell wall in bacteria. The main component of cell wall in bacteria is peptidoglycan. Okay, the important component which makes the cell wall in bacteria is peptidoglycan. See, depends upon the composition, the thickness of peptidoglycan. The bacteria are classified into gram-positive bacteria and gram-negative bacteria. Okay, the bacteria are classified into gram-positive and gram-negative. Okay, so look at that. So this is the plasma membrane, you can see, and above the plasma membrane, surrounding the plasma membrane, if there is a thick, okay, very thick peptidoglycan is there, then it is called what? Gram-positive bacteria. And look at that, the peptidoglycan, the blue portion, which is so thin, okay, which is so thin, then it is called what? Gram-negative bacteria. So, cell wall in bacteria is made up of peptidoglycan. If the peptidoglycan is so thick, that can be categorized as gram-positive bacteria. If the peptidoglycan is thin, then it can be called as gram-negative bacteria. Okay, if possible, later we will be doing the practicals to know, identify how like the gram-positive bacteria and gram-negative bacteria. There is a procedure called Gram staining procedure. Okay, a scientist called Christian Gram. So he is the one who established the method to classify the organisms, especially bacteria, based on the type of cell wall it possesses. So that is the broad way, important way of classifying bacteria. Presence of thick cell wall or thin cell wall, gram-positive or gram-negative. Okay, fine. And next, coming to the cell wall in fungi. The main component, or you can say this is the plasma membrane. The main component are chitin. Okay, cell wall in fungi are made up of what? Chitin. Okay, of course, other manoproteins, glucans, other polysaccharide molecules are also present as the component of fungal cell wall. Okay, which one? Glucans. See, like important component of that actually. Beta glucans. Okay, like alpha glucans, beta glucans. Okay, so it is called the beta glucans. But primary component is the chitin. Okay, so bacterial cell wall is made up of. Can you tell me the components of all kind of organisms? Bacterial cell wall is made up of peptidoglycan. Fungal cell wall is made up of chitin. Major component is chitin. Plant cell wall is made up of Primary cell wall is made up of cellulose, hemicellulose, and pectin. Secondary cell wall is made up of lignin. Okay, fine. Lignin and suberin. Right. So, and next. So this is about the plasma membrane. Okay. See, this is the cell. You can see, and this is like the cytoplasm. Okay, cytoplasm, and which is completely covered by the phospholipid bilayer. Okay, it is covered by what? The phospholipid bilayer, right? And can you see what are the structures here? What kind of protein it is? Integrated membrane protein, intrinsic membrane protein. And these proteins which are present on the surface of the cell. This is the outer surface and this is the inner surface. So these proteins are called what? Extrinsic protein or peripheral protein. Okay, membrane integrated. You can see these proteins are integrated in the membrane, and these proteins are present on the surface of the membrane. They may be present outside or it may be present inside. Okay, which may be facing outer environment of the cell or cytoplasm of the cell. So both are peripheral membrane proteins. Okay, good. So this is the basic model, and you can look at how these components makes the cell. Okay, see the phospholipid bilayer. We saw yesterday, right? The amphipathic nature of the phospholipid bilayer. What is called amphipathic nature? Yeah, this is the head, the dark blue color at the head of the phospholipid molecule, and this is the tail. Head is hydrophilic, tail is hydrophobic. Okay, so look at that. The molecules, phospholipid molecules, are present in two layers, facing outside as well as inside. Right. So that's why the model of the cell membrane is called what? Phospholipid bilayer. Okay, model. Okay, also it is called fluid mosaic model. Right? It is called fluid mosaic model. See, look at that. These lipid molecules positions. For example, this molecule can go here. This molecule can come here. Yeah. Okay. Like they can change its position also. Okay, they can change. Not inversion or whatever, but they can change. Okay, here to here, just to facilitate the movement of substances in or out. Okay, so majoritarily the cell membrane is made up of the phospholipid bilayer, the blue one, and you can see the red portion. Okay, like so this is what the channel proteins. Okay, protein molecules, the other important component of the phospholipid bilayer means the cell membrane. And you can see there are like two, as we saw here, two type of protein molecules. You can see here like the membrane integrated protein and also the peripheral protein. Okay, the third important type of molecules are called what? The cholesterol. Can you see the yellow portion here? Okay, this yellow portions are what? Cholesterol molecule. Okay, like, and also you can see like there are two type of carbohydrate molecules. See, either these carbohydrate molecules are attached to the brown color protein portion. If the carbohydrates are attached to the protein portion, they are called what? Glycoprotein. Okay, glycoprotein, because they are attached to the protein molecule. Also, they are called what? You can say lipid molecule. They can be attached to the lipid molecules. Okay, if they are attached to the lipid molecules, then they are called what? Glycolipid. Okay, so what are all the four important components? You can see you phospholipid bilayer, and then protein molecules, and then cholesterol, and then carbohydrates. Okay, so thus the cell membrane is composed of all these four molecules. Okay, right. And so this is about the transport across cell membrane. Okay, Do you remember the name of the mechanisms? To look at that oxygen, maybe from higher concentration to lower concentration, it can enter. What is the name of the mechanism? Passive diffusion. Okay, so high concentration to low concentration. And see, through the phospholipid bilayer itself, the oxygen can enter. Okay, through the phospholipid bilayer itself, the oxygen can enter. Is it clear? So this is passive diffusion, high concentration to low concentration. Passive transport system involves motion of substances from high concentration towards low concentration. Okay, and no big energy is there. Just like that, the transport can be facilitated occur. Okay, and look at that. This is diffusion through channel. Look at that. The ions are not permeable to the cell membrane. Okay, phospholipid bilayer cannot allow ion molecules to be passing through. Thus, they need a facilitator for its transportation, right? So, the ion molecules can be transported through channels, ion channels. Okay, ion molecules can be transported through ion channels. Okay, what is the ion molecule here? What is the name of this ion? Sodium, Na+, sodium. Can be transported through sodium channel, sodium aqua channel. Okay, and look at that. What kind of transport it is? Since it is diffusion, what kind of transport it is? Passive transport. So high concentration to low concentration. Always in extracellular fluid, outside the cells fluid, sodium is in high concentration. Okay, thus sodium can passively diffuse through sodium channel. Can you understand? Fine. See, there are, you can see this channel is, different type of channels are present in the cell membrane. So this channel is called actually ion channel, also called the open channel, open gated channel. What is called open gated channel? They are always open, and the structure of the ion channel do not change by the entry or exit of any substances. It is fixed. The shape of the structure, the channel is actually fixed. Okay, this always open also. This is open channel. Some channels are actually called what? Closed channels or gated channels. Gated channel in the sense, some channels can allow only, for example, voltage gated channel. They are closed all the times. Only voltage is reaching the channel. That time it will open. Otherwise, it will not open. Okay. So what are the two type of channels? Open channels and gated channels. Okay, closed or open channels. Okay, fine. So look at that. There are channels are present in the cell membrane to facilitate transport, pumps to facilitate transport. Also, transporter system is available in the cell and which is very selective. For example, this is glucose transporter system. Okay, glucose transporter system. Again made up of protein channels only. Membrane integrated protein channel only. But selectively permeable. What is called selectively permeable? This channel, yeah, can allow only glucose. If it is a glucose transporter system, it can transport only glucose molecule. Okay, and the glucose also need a carrier protein to enter. What is called a carrier protein? This glucose has to attach with another protein molecule. That attachment only can make, okay, that carrier cargo protein moves the glucose from outside to inside. Yeah, clear? So that is called what? Transporter system based movement of substances, right? And then these two are actually, all these three, either through phospholipid bilayer or through channel, open gated channel, or through specific transporter system, Anyway, all these substances are high in its external environment and low in its internal environment, thus they can passively diffuse. Okay, so this is passive transport. Look at here. Okay, you can see one pump. This pump is actually called what? Sodium potassium pump. Okay, sodium potassium pump. See, what you can understand from this is, see, look at that. What is ECF? What is ICF? Intracellular fluid and extracellular fluid. See, in a mammalian system, in our system, always sodium is present in high concentration. You can see high concentration of sodium is present outside. Low concentration of sodium present inside. So, what can occur? Sodium can generally high to low can come. But here, this is the pump. Okay, you can see the symbol. The function of the pump is actually from low concentration, sodium should be pumped out to the high concentration. Okay. Similarly, like that, in the intracellular fluid, potassium present in high concentration. In the extracellular fluid, potassium is present in low concentration. Okay. In ECF, potassium is low. In ICF, potassium is high. In ECF, sodium is high. In ICF, sodium is low. Okay, so a viable cell always maintain non-equilibrium steady state. What is called non-equilibrium steady state? Always, even though channels are present, pumps are present, always sodium will be present in high concentration outside, low inside. Potassium high inside, low outside. Right? So this is non-equilibrium steady state. This is favored by the pump. For example, through sodium channel, look at that. Many sodium can easily diffuse. Okay, passively it can diffuse. That sodium has to be, look at that, sent out, pumped out. The excess sodium which is coming in must be pumped out with the help of what? Sodium potassium pump. And you can see this mechanism is actually called what? Antiport mechanism. What is called antiport mechanism? Against each molecule, movement occurs against each of the molecule. In the sense, one molecule goes out, one molecule comes in. Okay, entry as well as exit. Okay, sodium can exit through this pump and potassium can enter through this pump. Can you understand? Sodium and potassium, both of the ions uses this transport system, pumping system. Okay, thus the homeostasis is achieved. In the sense, normal concentration of sodium outside, normal concentration of potassium inside is achieved. Okay, for this, what is required? ATP is required. Without energy, the pump cannot function. So ATP is the energy currency. Okay, so with the utilization of energy, sodium, excess sodium inside is pumped out. Excess potassium outside is pumped in. Okay, so this transport is called what? Active transport. Okay, can you understand the function of cell membrane? How it regulates active transport as well as passive transport. And the second important mechanism of cell membrane transport is osmosis. What is osmosis? Yeah, diffusion of water. Okay, but see, water or the diffusion, like there are two compartments, right? You can say outside the cell, inside the cell. Is enhanced by what? It is influenced by what? Solute concentration. Can you see the goody substances? Here it is more. Okay, here it is more, and here it is less. Solute concentration is less. Okay, but water in the initial stage it is equal in both of the compartment. Can you see? Water in both of the compartment is equal. This is, you can say, A compartment, B compartment, or left, right. Water is equal, but what actually varies? The solute concentration, chemical concentration. See, the kuti bubble represents the solute concentration. See, in the initial time, here solute concentration is more, solute concentration is less. So water will be having more potential here. So water can move from this compartment to this compartment, right? Okay. So high water potential to low water potential. Okay. After few minutes, look at that. Okay. The solute, look at that, what the solute will do from high concentration to low concentration, vice versa, right? Water can move from here to here, solute can move from here to here. Can you understand? So because of that, what you can see in compartment B, you can see more water diffused. Here, you can see the solute concentration, most of the solute molecules from high concentration to low concentration. Here, they move. Okay, so the movement of water between two compartments, maybe in and out of the cell, okay, in and out of the cell, is influenced by the chemical concentration. Is it clear? Depends upon the concentration of the solution. Movement of water is influenced. Is it clear? So, if water moves out to in, what it is called? Endosmosis. What is endosmosis? Movement of water from outside to inside. What is exosmosis? Inside to outside. Okay? Fine. Good. And then there are other bulk transport mechanisms. Actually, there are 4 processes which we saw. Exocytosis, endocytosis. What is the exocytosis? See, look at that. This is cytoplasm of the cell. Cytoplasm of the cell. This is extracellular fluid. See, the protein molecules which are made or transported through vesicles. Okay. Look at that. We will be discussing the functions of like endoplasmic reticulum, Golgi apparatus and all. See, the proteins are made inside the cell. The green portion represents the protein and they are covered by the vesicle, membrane bound vesicles. These vesicles, what is the function of these vesicles? To protect the protein from its internal digestive enzymes and to transport the protein. Okay, protection and transportation, right? So the green color molecules represent the protein molecule transported through the vesicles from inside to outside. Okay, this mechanism is called what? Exocytosis. And you can, endocytosis means movement of substances from outside to inside. Okay, and there are 2 important processes called phagocytosis, cell eating process, pinocytosis, cell drinking process. See, the solid substances, for example, a bacterium is there. Okay, or any other food substances are just engulfed by the cell and they along the membrane. You can see along the portion of the membrane, it is transported inside. Is it clear? So this process is called what? Phagocytosis, cell eating process. And then cell drinking process is pinocytosis. Pinocytosis means in the sense, not the solid particles, but the little oil droplets or whatever. Fluid substances can be taken, same mechanisms. Okay, instead of the solid particles, the fluid particle can enter. So that is called what? Pinocytosis. So what are all the different kind of cell membrane transport mechanism? Passive diffusion, facilitated diffusion, transporter-based diffusion, active transport by pumping. And then osmosis, movement of water, 2 types, endosmosis and exosmosis. And then here bulk transport, exocytosis, from cytoplasm to the external environment through vesicles. Endocytosis, solid particles entry through phagocytosis, liquid particles entry through pinocytosis, cell eating or cell drinking process. So these are all the different mechanisms which enhances the movement of substances in and out of the cell through its membrane, right? And now, so we saw cell wall, and then cell membrane. Now, what is protoplasm? Yeah, the cytoplasm, including the nucleus, is called what? Protoplasm. Okay, what is cytoplasm? Jelly-like substances without the nucleus, excluding the nucleus is cytoplasm. Okay, fine. This cytoplasm only, okay, like organized in animal cells and all eukaryotic cells, animal cell as well as in plant cells. Most of the organelles, as we saw, are located inside the cell membrane. Okay, in the cytoplasm. In the protoplasm only, all of the organelles, almost most of the organelles are present. Okay, fine. So we are going to discuss about each and every organelle, their structure, their primary function. Okay, what are all we are going to discuss? Different organelles present, their structure, their primary function. Okay. First, let us start with the nucleus. Okay. Nucleus is present only in eukaryotic cell. Okay. The presence of double membrane structure. Okay. Membrane bound. Nucleus is a membrane bound organelle. Okay. Membrane bound. We can see two membrane, outer membrane and inner membrane. Two membranes are present in the nucleus. Okay. Inside nucleus, what is present? Yeah, the DNA, the nucleolus 150 organelle, and then you have chromatin fibers. Okay. This whole area is called what? Nucleoplasm. Okay. So, nucleus is the chief organelle, okay, which is present inside all eukaryotic cells. Maybe some cell lacks nucleus also. Some eukaryotic cells lack nucleus also. Can you give me some example of cells which lacks nucleus? Eukaryotes do not have nucleus. Fine. Are there any eukaryotic cell which lacks nucleus? Yeah, the mature red blood cells, mammalian mature red blood cells lacks nucleus. Okay, fine. Look at it. So, the double membrane, look at the structure, the double membrane inside nucleolus, and then you can have the chromatin fibers, chromosomes in linear form. Okay, DNA in linear form is called what? The chromatin fiber is present, okay, internally. And then if you look into the outside of the cell membrane, see, nucleus is a double membrane structure. The extension of the outer membrane. Can you see the extension of the outer membrane is again forms what organelle? Endoplasmic reticulum. Okay, the extension of the nuclear membrane forms endoplasmic reticulum. Okay, and there are of course two type of endoplasmic reticulum. What are? Rough and smooth. Rough means they are studded with what? Ribosomes. Okay, fine, good. So look at the important component of nucleus, and each and everything are having its own function. For example, what is the function of nuclear envelope? Yeah, nuclear envelopes protects the internal content, also the external content. No unnecessary substances can enter through nucleus. Okay, so also the inner content, just like that, it cannot pass through. Okay, like only it can pass through what? Nucleopores. It's in the nuclear membrane. You can see pores. These pores are called what? Nuclear pores. And another important thing is this nuclear pores alone is not necessary, means not can guide the molecules in and out. Even though nuclear pores are there, the molecules needs signals to pass through the nuclear pores. They are called NLS, means nuclear localization signals. What are they called? Nuclear localization signals. Okay, through that signals only the molecules can pass, get in or get out through nucleopores. Okay, very complexly and organizedly regulated. The movement of substances in and out of the nucleus is more regulated than in and out of the cell. Most of the molecule can easily enter into the cell, but cannot easily enter into the nucleus. It is even more protected well. Okay? Fine. So what is called nucleoplasm we saw. Chromatin. What is the difference between DNA, chromosomes, and chromatin fiber? See, DNA. Yesterday we saw DNA is 2 meter in length, right? The DNA is, yeah, it's actually 2 meter. Okay, one DNA is actually 2 meter in length. Yeah, 2 meter. Okay, DNA is 2 meter, but it is also linear in structure. It is double stranded and which is linear in structure. Okay, but how it is present inside? The DNA, linear structure of DNA is actually like coiled. Okay, we saw it is coiled with histone proteins. Histone octamer forms. Okay, histone octamer forms and which coils the DNA. Okay, like that DNA nucleosome, DNA nucleosome, DNA nucleosome, DNA nucleosome. Okay, nucleosome means what is called nucleosome. Okay, nucleosome. Okay, when the double stranded negatively charged DNA is packed with histone positively charged histone molecule. That structure is called what? Nucleosome. When linear DNA is negatively charged, packed around positively charged histone proteins, that structure is called what? Nucleosome. Okay, the structure is called what? Nucleosome. And you can see DNA nucleosome. Nucleosome. This structure is called what? Chromatin fiber. Chromatin fiber. And this chromatin fiber again condense. And this is called what? Chromosome. Can you understand? Is it clear? Okay. What is this? DNA. Okay. They're called histone proteins. Okay. Then this DNA. This is called what? And nucleosome. Okay. It's called what? Chromatin fiber. And chromosome. Is it clear? Fine. Now, just the theory of that. It is like a double membrane structure, and you can see has pores for the movement of substances inside and outside of the nucleus. It contains chromosomes. Okay. Generally, the DNA is present in chromatin fiber form. It is condensed. Okay. It is condensed only during the cell division process. Okay. When the cell is about to divide, that time only the linear structure chromatin fiber contains and forms chromosomes. Okay, and chromosomes are composed of positively charged histone proteins and negatively charged DNA molecule. Okay, fine. The functional unit, the active portion of the chromosome is called what? Genes. Genes only determines all the characters. Is it clear? Fine. The functions of nucleus. It plays an important role in cellular reproduction. Every cell can multiply only if the nuclear content multiplies. Means the DNA multiplies. Then only the cell can multiply. Okay? Thus, also genetic information is stored in the nucleus only. Okay. Any species can produce its progeny only with the genetic information which is stored in the nucleus. Is it clear? Any doubt? Yeah. Fine. So, main function is nucleus stores the genetic information and plays a crucial role in heredity, transfer of the hereditary information from parents to offspring. For example, human being produces gametes. Okay, in sperm cell. For example, in male, sperm cells are produced. Sperm cells contains what? DNA. Okay, the nucleus, the DNA, half of the genetic information. In female, egg cells are produced. Egg cell contains what? Again, DNA, the chromosomes. Okay, in the process of fertilization, both fuses, zygote forms, the organisms develops. So, the main function of nucleus, reproduction, heredity, storage of genetic information, and making the information so stable. Okay, very stable. Look at it. Our father and mother, they are having two eyes, one nose, two hair. Look at our body pattern, organ, because the information is like that only. If they, while transferring the genetic information, if they does any change, if any change occurs, how we will be? We may be missing our hair. We may be missing our nose. Our nose can be positioned on top of your head. Can you understand? So the developmental disorders. See, look at that. The function of nucleus is it has to store as well as protect, okay, from the stress or any kind of damages. Is it clear? So that is very, very important. It has to transfer as well as in a safer way. As such, what is there in the parent, as such, it has to be transferred to the progeny. So very, very important role the nucleus does. Okay, fine. Does the prokaryotes have nucleus? No. Prokaryotes do not have well-defined nucleus, but the DNA which is present inside the prokaryotic cell is called what? Naked DNA. Okay, so naked. What is naked? No nuclear membrane is present. Okay, but it is present as such. Just like that, the coiled DNA is present in the prokaryotes. Is it clear? So, hence it is called what? Naked DNA. That region is called what? Nucleus. Okay, nucleus is the region of DNA which is present in the prokaryotes. Okay, that's why they are called what? They are called what? Prokaryotes. Pro means what? Primitive. Karyon means nucleus. Okay. No defined nucleus. That's why they are called prokaryotes. Eukaryotes also we have true nucleus. Organisms with true nucleus. The next organelle is what? Most of yes, because nucleus contains DNA. All the component of the cell arise from DNA. Okay, most of the proteins, DNA has gene. Gene is expressed and proteins are produced. Okay, something is called a central dogma. They call it as central dogma. What is central dogma? like the nucleus possesses double stranded DNA molecule. Okay, this results in the production of what? Single stranded RNA molecule. This RNA molecule has to be transported to the cytoplasm. In the cytoplasm, there are, you can see, ribosomes are present. These ribosomes uses this RNA is actually called what? mRNA. This RNA is called what? mRNA. Okay, mRNA. It brings the message. This message is translated into protein. Okay, that protein, if you look into the component of ribosomes, what is the structure of ribosomes? Proteins are there. There are two types of protein molecules. Okay, and inside that RNA is there. Okay, ribosomes are protein and RNA based component. Okay, so again, this protein can be synthesized from the information DNA which is present inside. So nucleus plays a crucial role in production of all type of protein molecules. Is it clear? Yeah, major component of like all the protein, most of the proteins are synthesized with the help of nucleus. Okay, nucleus only controls all the activity. Okay, of a cell. Every cell function is regulated by the nucleus. Okay, fine. So look at this. Yes. Can you be little more audible? Uh, nuclear lamina. Okay. See, you're asking about this. Okay. Yes, what is nuclear lamina? You're asking. See, a thin layer, okay, which is present inside the nucleus is called what? Nuclear lamina. A thin layer which is present inside the nucleus. See, pores are there now, but still there is a thin layer which protects the contents moving in or out, just like that. Okay, a thin layer which is called nuclear lamina. Okay, have you wherever any other places you heard this lamina? Yeah, lamination or middle lamina. Two cells are there, cell wall is there, but there is a thin layer, protective layer. We call this middle lamella. Okay, so like that, inside that there is a thin layer that is called lamina. Good. Okay. And coming to endoplasmic reticulum. Okay, it is, you can see a cup or disc. Okay, cup, sac, you can say. Cup like structure, disc like structure, or sac like structure. Okay, which is the extension of the cell nuclear membrane, outer extension of the nuclear membrane, which forms the endoplasmic reticulum. Which forms the endoplasmic reticulum. What are all the two type of endoplasmic reticulum? Yeah. If the endoplasmic reticulum is studded with ribosomes, it is called what? Rough endoplasmic reticulum. If it is not studded with, presented with the ribosomes, it is called smooth endoplasmic reticulum. In general, what is the function of rough endoplasmic reticulum? Synthesis of protein, because the ribosomes which is present in the membrane produces protein. When the protein is produced here, it enters into the lumen of the endoplasmic reticulum. It is double membrane structure, and inside that, okay, inside that which is called, that area is called what? Lumen of the endoplasmic reticulum. Okay, which is called what? Lumen of the endoplasmic reticulum. Okay, lumen of the endoplasmic reticulum. Here, assembly of protein occurs. What is called assembly of protein? See, what are polypeptides and proteins? A protein, listen, a protein may be made up of what? Many polypeptides, many single layer like single peptide chains makes a protein. Which is the smallest protein in animal body? Insulin. Okay, we have protein. Smallest protein we call it as insulin. Okay, know the function of insulin. Insulin is having, what is the monomer which forms protein molecule? What is the monomer which forms protein molecule? Monomer, protein is a polymer, which is made up of what monomer? Amino acids. Amino acids are the monomers